All right, so we're going to go ahead and get started with Unit 1, Section 1, which is going to be over learning targets 1 through 5, if I'm not mistaken, on your learning target list, which is basically an introduction, into agribiology, and also a review or introduction, whatever it may be for you, of the scientific method. So what is agribiology? Well, agribiology is a laboratory-based science course. The course emphasizes detailed knowledge of the biological principles of molecular and cellular aspects of living things, structure and function of agricultural plants and animals, genetics, physiology, plant and animal diversity, and principles of classification, ecological relationships, and animal behavior. So why is it important? Well, first off, biology is one of the most fundamental processes of life. Biology is the study of life and all things living. So it's important because we're living, and most things that we interact with or rely on are also living. So we need to understand how they operate. It is important to understand not only how we function, but also the environment around us as well. It is my intention for you to leave this class with a greater appreciation of agriculture, biology, and to give you positive ideas for career in the field of agriculture and biology. So what are the uses? Study methods for increasing the productivity of cultured plants, domesticated animals, and beneficial microorganisms. It incorporates advances in genetics, physiology, biochemistry, biophysics, and molecular biology. So, one of the main uses is finding ways to make things more efficient and less impactful on the environment, while accomplishing a goal faster. So how have we come to know so much about science? Well, it's kind of twofold. Number one, you solve problems, and those problems show up when people asking questions. So you solve problems and answer questions. This is the fundamental aspect of all science. And you see a question, or you have a question, you formulate a question, somebody asks a question, then you answer that question. You solve problems. If you're growing something that takes too long, if the growing season is too short, but the product you're wanting to grow is too long and doesn't fit in that growing season, then that's a problem. How can you fix that problem? What can you do to maybe produce that faster, or is it even possible? So what do scientists use to do this? Well, it stems to the scientific method. And you probably had the scientific method before now. Like I tell all my classes, if you go to the internet and you type in scientific method, you're going to get a lot of different diagrams, a lot of different steps, but it all comes back to the same process at the bones of it. So how can you understand the scientific method, or how can the scientific method be useful to you? So you need to learn how to formulate strategies to answer questions you will encounter in your own scientific studies or daily lives. I like to think that all of us, including you, do this on a daily basis without us even realizing that we are thinking like a scientist. We're faced with problems every day. You're faced with problems every day, and you solve those problems. Some take longer to solve than others. Some are solved fairly quickly, and it's out of habit, but that habit was formulated and started somewhere. That's a scientific process. You're being presented with a challenge, and you're overcoming that challenge, no matter how large or small that challenge may be. Or you may ask questions throughout your daily lives. Maybe at some point today you saw something and it made you say, I wonder why that happened. And then you start thinking about it. That's a scientific process. That's using the scientific method, far be it, or although it may be a, relatively elementary example, but it's an example nonetheless. Anytime you are asking yourself or someone else a question and then you work to figure that question out, regardless of how much effort you put into it, that's a scientific method. So again, ask questions. Questions are great. questions breed knowledge. The only bad questions are questions that are left unasked. I truly believe that. Ask the question. Uh, Whether it's you're noticing something, or even if you're in a class, or if you're going through these videos and you're saying this just does not make sense to me. I need more. Ask the question. I would bet money that somebody else has the same question. All right, so after you ask the question, the next step is collect data. All right, so how do you collect data? Well, the best way is to observe, or one way is to observe. Observe what? Well, you observe what's going on. Okay, is it putting off light? Is it putting off smoke? Is there sound coming from whatever it is you're doing? Whatever you saw, anything that you can, pick up with your senses is typically observing. Measure, okay, you take measurements, and this is getting into two different types of data: quantitative and qualitative. Right? Quantitative, quantity, is the amount or a number amount of data or pieces. Qualitative, quality, is you're describing something. Okay, you're describing a product. Is it rotten? Is it soft? Is it what color is it? Does it look healthy? Does it look sickly? Those type of things. It could be the sample size. Okay, you're looking at the sample size to collect data. Is it a large or small sample size? Is it random or is it fixed? Those are all important. And then most importantly, there at the bottom is you organize your data, and we're going to work on that in this class. Data tables are important. The average Joe Blow needs to be able to pick up your data and understand what it's saying. Again, numbers are just numbers without any type of meaning to them. I don't know, if you just turned into me a notebook paper with a bunch of numbers wrote down on it in a row, I don't know if you wrote down the class's shoe size or how many, coins every person had in their pocket. I don't know what those numbers mean. So it's up to us as scientists, or up to us as individuals, to put meaning to those numbers, and that is done with data tables and charts or graphs, or any way that we can organize that data to make it more presentable and reader friendly, so to speak. So after that data is collected, then you form a hypothesis. So a hypothesis, I like to think of a hypothesis as an educated guess. You're using data that is previously collected, or research that is previously done about whatever your observation in question was, okay, to formulate an educated guess. Right? So create a statement that explains the observations and can be tested. So you've seen your phenomena or your event that made you ask a question. Okay, you ask the question, then you research or collect previously known data about your event that caused your question. Now, using that knowledge, you say, okay, I think this happened because la 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 la. All right, it's a guess to answer your question. That's a hypothesis. Okay, so a prediction is a statement that predicts in advance the results that will come from testing a hypothesis. Usually, it's an if-then statement. So it could be, if whatever your hypothesis is true, then I predict this, and they go one in this, one in hand, or however that saying goes. They relate to one another. So then you carry out your experiment. Okay, you gather data under controlled conditions. You have a control group versus an experimental group. And it's important to know the difference between the two. A control group is something that is not being, or not something, I should not use that word. It is a group that is not being manipulated. The experimental group is the one that's being manipulated with variables. Okay, so let's talk about this. Let's think about in terms of if we were doing a trial run on different types of plant fertilizers as it relates to growth. Okay, so let's say we're testing three different types of generic plant fertilizers that you can get at Lowe's or Home Depot, and the fourth group is going to be our control group. That control group is going to get nothing but water. That's it. The bare minimum for life, which is water, and then whatever nutrients are in the growth media that we've planted it in. Right? And the other three groups are going to get a fertilizer. And we're testing to see which fertilizer is better, but we need a measuring stick. We need to know what a plant's going to grow on its own without fertilizer. That's our control group. Okay? So that's a real rough example of the difference between a control group and an experimental group. So after you do your experiment and you've collected your data from your experiment, then you analyze that data and you come up with conclusions that does that data support your hypothesis and your prediction? Okay. If it does, great. If it doesn't, great. Yes, I said that right. If it doesn't, great. Okay. One thing that we'll talk about in this class, a lot of discoveries in science came from failed experiments or by accident. And that's the great thing. Let's say that your experiment did not go like you wanted it to do. It did not end the way you thought it would end. That doesn't mean it's a failure. You can always learn from those things. What are you going to adjust? Maybe whatever the end result was better than what you originally thought it may have been. And maybe it's going to open up another door, maybe make you ask another question, which is going to lead to more knowledge. You see where I'm going with this. Okay. While you're analyzing the data, you look at what the variables were. Okay. What did you manipulate? What was not manipulated? How did it affect things or how did it change things? What was your control group? How did it relate to the control group? What was the temperature or the amount of sunlight or the humidity, the amount of water? Again, there's a lot of different variables. Are there sources of error? 99.9999999% of the time, the answer to this question is yes. And you want to know why? It's called human error. We humans are not perfect. We make mistakes, whether it be in our measurement techniques, whether it be in our administration techniques, whether it be in our the administering of the variables for the experimental group. Maybe the timing was off. There is always going to be at least a small amount of error in experiments. Again, now make the case that sometimes there may not be based off what you're doing, but there still could be error in your note taking, in your data taking, in your data analyzing, all of those things. So, are there sources of error? More than likely, yes, and we need to know what those are. We need to know where the variations may possibly come from. So after that, then you draw a conclusion. You shed light on something previously not understood, hopefully, one way or the other. Okay. Again, no data is bad data. No end result of an experiment is a bad end result. Okay. You can always learn something. You can always take that knowledge to apply it to something else. You're going to have to redo that experiment. And there's no such thing as a one and done when it comes to experiments or when you're trying to figure something out. You do it multiple times and you get better every time you do it. Then you can construct a model. Okay, so a model is an explanation supported by the data that you collected. So this model can be visual, verbal, or mathematical. Okay, most of the time people tend to go with a visual model that is presented verbally. Sometimes those visual models do have some math that goes with it. So, sometimes the best models are a combination of all the three. All right, make an inference. So this term inference, okay, is a conclusion based on facts rather than observations. Inferences can't always be tested, okay? So an inference is a conclusion based on facts rather than observations. Then you form a theory. This is not a wild guess or a hypothesis. Theories are formed after several hypotheses have been tested and supported by experimental evidence. It's a broad, comprehensive statement of what is thought to be true. A theory is not an educated guess. A theory has data to back it up. Okay, that's the difference between a theory and a hypothesis. You start with a hypothesis and you work with that hypothesis multiple times over and over and over and over and over. You collect a lot of data. Your data is pretty consistent. Now you're ready to say, okay, this might be a theory. Here's my theory. Here's my theory for this question, and here's the data that backs it up. So again, the steps of the scientific method, okay, a little bit different than what you may find online, but it's still pretty good. Number one, ask a question. Number two, collect data or previously known information about your question. Number three, form a hypothesis. Number 4, make a prediction. Number 5, conduct your experiment. Number 6, analyze the data collected from your experiment. Number 7, draw a conclusion. Number 8, construct a model. Number 9, make an inference. And last but not least, number 10, form a theory. And that's where we'll end today. Be sure you complete the question form that's found on your pacing guide. The link is there right underneath this video link. Answer those questions that go with this. And I hope you've had a great week. Be sure you get everything turned in this week, and we'll pick back up again with this next week.